Welcome everybody. This is Felix Drost from the Department for Computational Health of the Helmholtz-Zentrum Munich in Germany. It's a pleasure to present to you the project predicting T-cell activation for mutational epitopes. Let's start with a small introduction to T-cells. Imagine a virus in your body that at some point will infect a body cell. This virus there can produce protein, which will be broken down into small peptide sequences, so-called epitopes. These epitopes are then presented on the cell surface of the infected cell via the major histocompatibility complex, or MHC. A T-cell can now interact with this MHC via the T-cell receptor and either recognize or not recognize this given epitope. Upon activation, the T-cell will release cytotoxins that will kill the infected cell. After this activation, the T-cell will proliferate and with this specific T-cell receptor and thereby form immune memory. Studying T-cell receptors is a challenging task because they have a quite diverse amino acid sequence. So there has been estimated to exist 10 to the power of 20 different T-cell receptors in nature, and 10 to the power of 6 to 10 to the power of 7 different T-cell receptors exist at each time in each human individual. Further, the space of possible epitopes is also quite diverse. Therefore, many tools have been developed that can predict the activation of T-cell based on the epitope sequence on the one hand, and the CDR3 region on the other hand. That is the region marked in purple here, which is part of the T-cell receptor and in close contact to the epitope. So most of the binding activity should take part here. The problem is that these tools are mostly trained on datasets that do not contain much mutational data of epitopes. So they are not well characterized for mutations. However, imagine a disease with a mutated epitope. Due to a previous infection with this disease or vaccination, we have been exposed to the base epitopes and formed an immune response and immune memory towards this epitope. Now we want to predict, can we, with the same T cells, also detect the mutated epitope, or can the disease escape our immune system? Another case, imagine a tumor cell, which is only a small mutation based on our body cell. Can our body produce T cells that can recognize the tumor cell and fight it without fighting our own body cells? So as you can see, understanding these small mutational effects on an epitope level is a quite important task for understanding our immune system and for the development of immunotherapies. Therefore, we used a novel dataset which contains 21 T-cell receptor sequences from a naive repertoire and 16 from an educated repertoire. Educated in this sense means that the mice have been previously exposed to our base epitope that we are studying. As a base epitope, here we use the well-characterized epitope with the sequence synfecal. From this, we formed every single amino acid mutation that is possible. So at each position of this synfecal sequence, we exchanged the amino acid against all possible other 19 amino acids. That gives us in total 153 epitopes and 37 T-cell receptors, giving us approximately 5,500 samples. For each of these combinations, we measured the percentage of activated T-cells that can be seen in this heat map plot here. On the first glance, you can already see that the T-cells from the educated repertoire have a much higher activation, reaching up to 90% of all T-cells that are activated here, while for the naive repertoire, most of the cells, the T-cell receptor sequences are not activated. This is due to the fact that for the educated repertoire, there have already been an immune response against the base epitope. So these are all T-cell receptors that can recognize synfecal. So it's more likely that they can also recognize its mutations. On the other hand, you can see that in in an individual column, we can have values from 90% to 0%. This is only a mutation at one position, so you can see that one mutation alone can have the impact of completely destroying activation of T cells. In our first step, we performed dimensionality reduction on an epitope level. For this, we used the activation vector of all T cells as an input vector and used UMAP embedding. The yellow star indicates our base epitope syncytial, and we can already see in the color coding that the epitopes cluster based on the position where the exchange took place. We can identify different clusters. On the one hand, we have positions that are highly robust to exchanges, like position 2, 5, and 8. Here, the activation does not change whether we mutate at that position or not. On the other hand, we have positions like position 4, 6, and 7, where the T cells are very sensitive towards mutations at these positions. And then there are cases where it really depends which amino acids do we introduce at these positions, like position 1 and 3. From this, we already see that the position must have a quite important impact on T cell activation. Next, we tried to predict the percentage of activated T cells for individual T cell receptors. For that, we hold out several mutations and trained the model on the remaining one. So, starting from the T cell, from the epitope sequence, we obtained a numeric representation. So this gives us a biophysical, chemophysical properties of each amino acid and represents it in a numeric way. We then feed this to random forest predictors that will predict the percentage of activated T cells. On this slide, you can see the results from this predictor. We report the Spearman rank correlation coefficient, since the precise value of activation is highly dependent on the experimental setup. However, the ordering of the different mutations should not change on the setup, and so we use this rank coefficient. In the graph, you can see the results when trained on various data subsplit. So on the left one, you can see when we left one mutation out and predicted all the remaining one. On the first glance, we can see that the prediction is far better on the educated repertoire. Here we have a better mixture of activating and non-activating T cells, and we have also larger differences between the activation scores. So this might be a reason here. And from the different training sets, we can already have some insights. First, when we leave a whole amino acid out in the training set, so the model has not seen mutations with this amino acid, the model has still quite high performance. So this builds trust that our model actually uses this biological representation, because otherwise it would not be possible to generalize for unseen amino acid. Secondly, we can get away with training only on 25% of the training data and still have comparatively high performance. Thirdly, this does only work if we sample evenly across all positions. So if we do not show the model mutations at a specific position, the model performance will decrease quite drastically. In the next step, we ask ourselves, okay, we can generalize inside a T-cell receptor, but can we also generalize across different T-cell receptors? For that, we adapted the model so that we can also use the T-cell receptor, especially the CDR3 region of the T-cell receptor, as an input. And for that, we use the same numeric representation that we already used for the epitope. We further thresholded the activation score by using the threshold for in vivo recruitment of T cells, so that we have a binary classification problem predicting whether the T cell is activated or not, instead of predicting the precise activation score. On this slide, you can see the results for this training across T-cell receptors. Here we report the receiving operating characteristic curves, and especially the area under the curve. On the left, you can see the results for the educated repertoire, and here we have pretty high performance with an ROC of 0.9, which is pretty decent. On the other hand, for the naive repertoire, we only have a value of 0.7. So this shows us that we can actually predict quite well for high reactive T-cell receptors from either the educated and the naive repertoire. But on the other hand, we lack performance for the mid-reactive TCRs, so we would need more of these for training to have better performance here. This is quite intuitive because most of the high reactive T-cell receptors will recognize the epitope in a similar way. But however, there are many ways for this mid-reactive T-cell receptors to look like, so we would need a much broader coverage to actually be able to predict them well. Finally, we wanted to see what our model actually does. So for that, we on the one hand modeled the T-cell receptor and the epitope using TCR-PMHC models to obtain a three-dimensional representation of the action that you can see here on the right side. From this representation, we measured the atomic distances between the amino acids in the epitope towards the nearest position in the T-cell receptor. On the other hand, we performed permutation-based feature-important tests on our random forest model. That can be seen here on the left. So we randomly permutated the input and observed how much the performance of our model decreases. As a sanity check, we permutated the CDR3 region, and there the performance dropped by quite large margin. So that indicates that our model actually uses the CDR3 region for its prediction, which is a good sanity check here. We then compared the effects different epitope position has in our model with the three-dimensional representation. And we found out that the positions that are in close contact in the epitope to the T-cell receptors are those positions for which our model is very sensitive. On the other hand, the position further away, position 2, 5, and 8, that in literature are also reported as anchor positions. So they are used to fix the epitope in the MHC and do not play a big role in the interaction with the T-cell receptor. We can see that our model is quite robust at these positions. So combining this gives us good confidence that our model has actually learned valuable features, because it makes biologically sense that the model is robust and sensitive to these specific positions. To summarize, we predicted the T-cell activation towards mutational epitopes which have very similar amino acid sequences. We did this by using random forests, and we have quite high performance within and across T-cell receptor. We further validated these models by using a three-dimensional model and running feature-important tests. We hope that with these models we can help to study escape mechanism of diseases and to determine cross-reactivity of small mutations. In future work, we plan to reduce the data that we need for training these models by sampling in a smarter way, so that we can further reduce the amount of wet lab experiments needed to train these models, which can reduce the time and the cost. In the end, I want to thank everybody who contributed to this project, especially Emilio Dorigati for the method development, Adrian Straub and Philipp Hilgendorf for the data acquisition, and Kilian Schober, Dirk Busch and Benjamin Schubert for their very helpful supervision. Thank you.